Hello, everyone, my name is Xiao. I am glad to introduce our review published on iMeta, Salmonella host microbiota interactions, immunity, and metabolism. Salmonella infection exploits host inflammatory response, employs immune evasion strategies, and manipulates host metabolic pathways to facilitate its proliferation. Meanwhile, host defends against Salmonella infection primarily through the activation of both innate and adaptive immunity. Furthermore, the host gut microbiota modulates the immune responses and metabolic functions, thereby influencing the complex interplay between host and Salmonella. Salmonella invades host mediated by the Salmonella pathogenicity island encoded type 3 secretion system, which transfers bacterial effector proteins into intestinal epithelium. Effectors like SOAP, SOP-B, and CYP-A can disrupt the tight junctions and induces cytosis. Once invading the epithelium, Salmonella replicates in the plasm and causes intestinal inflammation. Inflammation stimulates the release of tetrathionate and nitrates, providing growth advantages as respiratory electron receptors. Inflammation also disrupts the gut microbial balance, helps Salmonella to utilize the gut microbial fermentation products. Besides, Salmonella can use immune evasion strategy to promote infection. After endocyted by macrophages, it can replicate within Salmonella-containing vacuole, further induce SCV acidification to evade host immunity. Additionally, SOP-B can break host autophagic defenses to mediate autophagy evasion. After endocyted by macrophages, infected macrophages exhibit increased glycolysis and suppressed TCA cycle. The former leads to the accumulation of glycolytic intermediates, such as 3-phosphoglycerate, pyruvates, and lactates. While inhibition of the TCA cycle reduces the key intermediates like citrate, potentially due to the transfer rate from TCA to lipid biosynthesis. In turn, the levels of succinate and itaconate increased. Elevated itaconate induces lysosomal generation by alkylating the transcription factor EB, leading to bacterial clearance. Succinate can upregulate HIF-1 alpha expression, leading to increased IL-1 beta production. When Salmonella infects the intestinal epithelium, the NAPE and LRC4 inflammasome rapidly recognizes the pathogen, triggers epithelial layer contraction, thus maintains the epithelial barrier integrity and prevents further invasion. Macrophages can also clear Salmonella through extracellular traps release, referred to as etoses. Dendritic cells produce B-cell activating factor to stimulate the expansion of mature B cells, and further Salmonella-specific IgM production. The adaptive immune response involves CD4 positive and CD8 positive T cell activation. The former plays a key role in persistent infection by producing interferon gamma, the latter have been shown to exhibit delayed expansion. During infection, Salmonella interacts with the gut microbiota through multi-ways, such as competing for nutrients and influencing host metabolism. For example, commensal E. coli compete for carbon sources with Salmonella, induce caloric restriction to mitigate infection. E. coli can also compete for oxygen to reduce Salmonella colonization. Besides, E. coli can enter Salmonella niche to compete for nitrate, thereby decrease Salmonella colonization. In addition, bacteroids Thetaeomicron secretes lipoprotein ZUS-B to binding siderophores, but the ZUS-B-bound enterobactin are utilized by Salmonella, allowing host nutritional immunity evasion. Salmonella affects host metabolism by disrupting gut microbial homeostasis and promoting oxidative metabolism. Butyric acid-producing Clostridium was eliminated from the gut, leading to increased epithelial oxygenation. This, in turn, drives the cytochrome BD2 oxidase-dependent Salmonella expansion through aerobic and nitrate respiration within the intestinal lumen. The PPAR gamma agonists can decrease host lactate production and synergize with regulatory T cells to maintain colonic hypoxia environment and intestinal homeostasis, representing a potential metabolic pathway target for the treatment of Salmonella infection. In conclusion, the current research sheds light on the intricate interactions between Salmonella, host, and gut microbiota in terms of immunity and metabolism. However, further research is needed to fully elucidate the immune and metabolic mechanisms underlying these interactions. By better understanding these processes, it is possible to regulate host metabolism to enhance immunity and better defend against pathogen infections.